Hello everyone, welcome to the second PU biology practical session. In this session, you are going to study the reproductive parts of a flower. So, flower consists of 4 whorls: calyx, corolla, androecium and gynoecium. And the flower consists of a very small stalk-like structure, and this stalk is said to be pedicel. And the tip of the pedicel is like this, slightly flattened, and this part is said to be thalamus. On the thalamus, other floral parts are present. So, flower consists of 4 whorls: calyx, corolla, androecium and gynoecium. So, coming to the first one, first whorl is said to be calyx. So, the calyx is the first whorl, consists of sepals. Sepals are very small needle-like structure, gives protection to the remaining floral parts when it is in bud condition. And then coming to the second whorl is said to be corolla, consists of petals. Petals are brightly colored structure and it attracts insects for pollination. So, calyx and corolla together is said to be non-essential whorls. Then coming to the important one, essential whorls, the third whorl is said to be androecium. So, androecium consists of male reproductive units, and these male reproductive units are said to be, what do you call, stamens. The function of these stamens is to produce pollen grains, and then the innermost whorl is said to be gynoecium. So, gynoecium consists of female reproductive units. These female reproductive units are said to be carpels. So, totally flower consists of 4 whorls: calyx, corolla, androecium, gynoecium. Calyx consists of sepals, corolla consists of petals, and these two are said to be non-essential whorls, and the third whorl is said to be androecium, which consists of male reproductive units are said to be stamens, and then innermost whorl, that is said to be gynoecium, also known as pistil, and this gynoecium consists of female reproductive structures are said to be carpels. Next, we will move on to essential whorls. The first whorl is said to be androecium. Androecium, it is the third whorl of a flower, consists of a male reproductive structures are said to be stamen. So, stamen is like this, elongated part is nothing but it is a filament, and then the terminal knob-like structures are said to be anthers, and between the anther and the filament there is a presence of a tissue that connects these two, that is said to be, what do you call, connective tissue. This is connective tissue. So, if there is a presence of 2 anther lobes, then it is said to be dithecous anther. If there is a presence of only 1 anther lobe, then it is said to be monothecous anther. Now, when we take a cross section like this or a transverse section like this, here you can see there is a presence of 4 chambers. 4 chambers, and here these are nothing but microsporangia, and each microsporangium is covered by 4 layers. Outermost is nothing but it is a single layer, and this layer is said to be epidermis. Inner to the epidermis there is a presence of one more layer, and this layer is said to be endothecium. And inner to this endothecium there is a presence of a multi-layered structure or a region, so that this layer is said to be middle layer. These three together mainly helps in, or it gives protection, and it also helps in dehiscence of pollen grains. And the innermost layer is very important, and the innermost layer is said to be tapetum. This is the tapetum. So, innermost is the tapetum, which contains more than one nucleus, and the cytoplasm is dense, and here it gives nutrition for the development of pollen grains. So, this is about the transverse section of Dithecus anther. So, when we take the transverse section of Dithecus anther, we can see there is a presence of 4 locules or the chambers or the microsporangia, and each microsporangium which consists of, or each microsporangium is surrounded by 4 layers: epidermis, endothecium, middle layer. These 3 gives protection, also helps in dehiscence of pollen grain, and the innermost layer is said to be tapetum, and it is a nutritive layer. Now, inside this here chamber there is a presence of a homogeneous cells, similar cells, and these cells are said to be sporogenous tissue, and each cell that is present in the sporogenous tissue now act as a pollen mother cell, so that it undergoes meiotic division. Then there will be a formation of microspore tetrad. That means each microspore finally gives rise to pollen grains. So, here there will be a development of number of pollen grains, and these pollen grains are dehisced or comes out through this opening. This is said to be stomium. So, this is about the first essential whorl, that is Androecium. So, Androecium consists of male reproductive units are said to be stamens, and each stamen consists of filament, anthers, connective. Inside the anther there will be a development of number of pollen grains, and these pollen grains are dehisced through the opening, that is said to be stomium. Coming to the next one, next is Gynoecium. Innermost whorl of a flower, essential whorl, that is Gynoecium, which consists of carpels or the female reproductive units. So, here this is the thalamus, and here the carpel consists of 3 parts. Basal box-like structure, this is said to be ovary, and this is an elongated part, and this is said to be style, and the terminal flattened sticky part, this is said to be stigma. So, inside the ovary there is a presence of a fertile tissue that is said to be placenta, to which there will be an attachment of ovules. So, these are ovules. So, if the gynoecium is composed of only one carpel, then it is said to be monocarpellary. If the gynoecium is made up of two carpels, then it is bicarpellary. If the gynoecium is made up of three carpels, tricarpellary. If it is made up of four, then it is tetracarpellary. If it is made up of five, then it is said to be pentacarpellary. Now, if the gynoecium is made up of more than one carpel and all the carpels which are fused, then it is said to be syncarpous. So, what is meant by syncarpous? All the carpels which are fused, then it is said to be syncarpous. If the carpels are free from one another, then it is said to be apocarpous. Carpels which are fused, carpels which are free from one another. So, gynoecium, it is an innermost whole of a flower or the essential part in a flower, which consists of a female reproductive units are said to be carpels. Each carpel consists of ovary, style, stigma. Inside the ovary there is a presence of ovules. Now, if the gynoecium is made up of many carpels and all the carpels which are fused, then it is syncarpous, and the carpels which are free, then it is said to be apocarpous. Now, coming to the arrangement of these ovules inside the ovary, that is said to be placentation. Next is placentation. It's an arrangement of ovules inside the ovary. So, when we take a transverse section of the ovary and observe under the microscope, we can see there is a presence of number of chambers or the locules, which indicates the number of carpels which are fused. So, what is meant by placentation? Arrangement of ovules inside the ovary. There are different types of placentation. The first one is a marginal. Marginal placentation. It is seen in P. So, here you can see number of ovules attached to the ventral margin of the ovary like this. Therefore, it is said to be, what do you call? It is a marginal placentation. So, marginal placentation means ovules are attached on the ventral margin of the ovary. You can see in this picture. So, here this is said to be, what do you call? Marginal placentation. Second type is axial placentation. Axial placentation, it is seen in hibiscus. That means here ovules are attached to the central axis with the septa. So, what is meant by this septa? These are the septa. That is nothing but, this is nothing but the two carpels which are fused here. That part is said to be septa. So, axial placentation, it is seen in hibiscus. So, here what happens? All the ovules are attached to the central axis. Therefore, it is said to be axial placentation. In this picture, you can see this is the ovules are attached in the central axis with the septa. That is said to be axial placentation. Third one is parietal. Parietal placentation, it is seen in cucumber. Here what happens? Like this. These are the fused margins. Here the ovules are present at the fused margin like this towards the periphery. Therefore, it is said to be parietal placentation. You can see in this picture what happens here. The ovules are present towards the periphery, but it is attached to the fused margin of the carpel. Therefore, it is said to be parietal placentation. Fourth one is free central placentation. Free central placentation. Here what happens? The ovules are present in the central axis without septa. Therefore, it is said to be, what do you call? It is free central. It is seen in primrose. You can see in this picture, this is the primrose. Ovules are attached to the central axis without septa because it is made up of only one carpel, that is monocarpellary. Coming to the last one is the basal placentation. Here, single ovule that is present at the base of the ovary. Therefore, it is said to be basal placentation. So, this is about the placentation. So, what is meant by placentation? It is an arrangement of ovules inside the ovary. There are different types of placentation. The first one is the marginal placentation. Ovules are attached to the ventral margin of the ovary. Therefore, it is marginal. It is seen in P. And the second one is an axial placentation. Ovules are attached in the central axis with the septa. Therefore, it is said to be axial placentation. Best example is hibiscus. And the parietal placentation, it is seen in cucumber. In this case, what happens? The ovules are present at the fused margin towards the periphery. Therefore, it is said to be parietal placentation. Free central, it is seen in primrose. Here what happens? The ovules are present at the central axis without septa because it is made up of only one carpel or monocarpellary. And the last one is the basal placentation, the single ovule that is attached at the base of the ovary. It is seen in sunflower. So, this is about the Androecium and Gynoecium. So, this is hibiscus flower. You can see from here to here, this part is the main axis and this is said to be peduncle, and very small part, this is said to be pedicel, and the tip of the pedicel is, this part is the thalamus. On the thalamus there is a presence of other floral parts. So, the green color structures, these are the calyx. So, this is the calyx, we have to remove this, and coming to the next, brightly colored structures are said to be petals. Here also I am removing all the petals because these two does not take part in sexual reproduction. Coming to the next one, these are the yellow structures. These are said to be Androecium, which consists of male reproductive structures are said to be stamens, and the terminal 5 very small parts, these parts are said to be stigma. So, now we have to remove Androecium like this. So, now this is ovary and elongated part is the style and the terminal part is the stigmatic surface. So, the carpel consists of 3 parts: ovary, style and stigma. So, now in the examination we will provide this part. This is the ovary. What you have to do is, you have to take a transverse section. Take a glass slide, add 1 drop of water, and then you have to take transverse section of the ovary like this. Blade is dipped like this in order to avoid air bubble, and hold it like this, and then you have to take transverse section like this. And then with the help of brush, you have to transfer this section on the slide. And then cover this with the cover glass. Like this. And then extra water is there, you have to wipe it with the help of blotting paper, and this is ready for observation. This is ready for observation. So, you have to take ovary, and then you have to take a transverse section with the help of blade, put it on a glass slide with 1 or 2 drops of water, cover this with a cover glass, and then you have to focus by using dissection microscope. This is the dissection microscope. Why we have to use this microscope? Because we have to observe whole mount, complete section and observe ಮಾಡಬೇಕು. ಅದಕ್ಕೋಸ್ಕರ we are using this microscope. It's very simple, and only one adjusting screw or a knob, only this one. What you have to do is, this is the mirror. First you have to adjust the mirror towards the light like this, and then you have to keep the slide and then by observing the eyepiece you have to move like this. Here you can see 5 chambers. So in the microscope you can see like this 5 chambers, which indicates the 5 carpels which are fused. So therefore you have to write pentacarpellary. 5 carpels which are fused, therefore it is pentacarpellary. And then you can see 5 locules, therefore it is pentalocular. Pentalocular. And then you can see like this, the ovules are present in the central axis with the septa, therefore it is said to be axile placentation. So in the exam you have to do this experiment, and then you have to write only 2 important points. One is pentalocular, axile placentation. That means the 5 carpels which are fused, therefore it is pentacarpellary. You can see 5 locules, therefore it is said to be pentalocular, and all the ovules are present in the central axis, therefore it is said to be axile placentation. So this is about the parts of a flower, which consists of androecium and gynoecium. Androecium consists of male reproductive structures are said to be stamens. So inside the anther there will be a synthesis or the development of pollen grains, and in case of gynoecium, gynoecium is also known as pistil. So gynoecium consists of female reproductive structures are said to be carpel. So each carpel consists of ovary, style and a stigma. Inside the ovary there is a presence of ovules, and these ovules are attached to the fertile tissue that is said to be placenta. So in the exam we will provide hibiscus ovary. You have to take a thin transverse section like this, and then you have to focus it by using dissection microscope, and then you have to write only 2 important points, that is pentacarpillary, pentalocular and axial placentation. Thank you.